So today we are going to talk about adrenal gland. Okay, now we will be restricting our discussion with respect to one of the hormones associated or secreted by adrenal gland, that is glucocorticoid. Okay, we'll actually now be talking about glucocorticoid chemistry. Clear? Glucocorticoid chemistry. Now when we are talking about glucocorticoid chemistry, you must understand this, that we are actually talking about one of the layer or two layers of adrenal cortex. Okay, now you must understand this at first, that adrenal glands are actually paired structures. Okay, they are paired structures. Okay, now listen with all your attention. Adrenal glands are actually paired structures, and suppose this is your kidney. Okay, and these adrenal glands are present above the kidney. That's why adrenal glands are also referred to as suprarenal glands. Okay, they are also referred to as suprarenal glands. Clear? They are also referred to as suprarenal glands. Okay, now important point here we need to understand is this, that when we are talking about adrenal gland or suprarenal gland, we are talking about actually two different glands. Okay, now listen, adrenal gland or suprarenal glands, they are actually combined into a single structure, but they have two different functions. Okay, now listen, that adrenal gland, suppose this is our adrenal gland. Okay, suppose this is our adrenal gland. Okay, suppose this is our adrenal gland. Now listen, now a very important point you need to understand is this outer portion is called adrenal cortex and this inner portion is called adrenal medulla. But there is another important aspect here. The cortex is actually surrounded by another structure called adrenal capsule. Okay, it is adrenal capsule. Okay, clear? Now, so this is a very important point you need to understand. Okay, clear? Now, so this is adrenal capsule and this is what? This whole thing is adrenal cortex, right? This is adrenal cortex. This is our what? Cortex. And this is what? Medulla. Okay, this is adrenal cortex, this is adrenal medulla. Okay, now listen with all your attention. This distinction between adrenal cortex and adrenal medulla is actually embryologically distinguished. Embryologically, adrenal medulla have come from neural ectoderm or neuroectodermal cells. Neuroectodermal cells, that means they have a neuroectodermal origin. Clear? They have a neuroectodermal origin. But adrenal cortical cells are derived from mesoderm. Okay, they are derived from mesoderm. Clear? They are derived from mesoderm. Okay, so adrenal cortex is derived from mesoderm and adrenal medulla is derived from neuroectodermal cells, or they have a neuroectodermal origin, or cortical cells have mesodermal origin. Clear? So these are two variants which have come together to form a single gland. So that's why they have a double origin. Now listen, a very important point, why I am talking about these divisions. Now listen, when we are talking about glucocorticoid chemistry specifically, now listen, adrenal cortex is absolutely and only associated with, okay, adrenal cortex is only associated with steroid hormone production, okay. They are associated with what? Associated with steroid hormone synthesis. Associated with steroid hormone synthesis. Clear? Steroid hormone synthesis. Clear? And adrenal medulla is associated with production of catecholamines. Okay, catecholamines. We have greatly discussed in details the catecholamines when we were talking about autonomic nervous system, right? Okay, clear? So many authors says that actually this adrenal medulla is nothing but sympathetic ganglion cells which have come together and form adrenal medulla. Clear? Okay. So the cortex is associated with steroid hormone synthesis, and medulla is absolutely associated with or only associated with what? Catecholamine synthesis. Clear? So from where we get our glucocorticoid? Okay. Now listen, glucocorticoid is one of the steroid hormone. Glucocorticoid is actually a class of steroid hormone, and under that class the most important steroid hormone is this, which is called cortisol. Clear? There are many, but we will talk about cortisol in details. Okay? Do you know why adrenal gland is important? Now listen, in the external environment of a cell, there are many ions which should be taken in so that it can actually live, right? Now, to maintain this homeostasis of ions and all other things like glucose and all other things, steroid hormones are required. Okay? That's why adrenal gland is a very, very important gland, and therefore adrenalectomized animals don't live longer. That means the animals from which adrenal gland has been taken, they don't live longer. In a clinical study it has been seen. Clear? Okay. So at first now we will be talking about the structure, in short the structure of or histology of adrenal gland. Clear? So now we will be talking about histology of adrenal gland. Clear? Now listen. Now listen. Adrenal cortex is divisible into three main zones. Number one is zona means zone, okay, zona glomerulosa, then zona fasciculata, then zona reticularis. Clear? Zona glomerulosa, zona fasciculata, and zona reticularis. How to remember this is very simple. Listen, try to remember the first alphabet, GFR. Okay, glomerular filtration rate, So let me draw the adrenal cortex here for you. Now listen, what do you mean glomerulosa? They look like glomeruli, surround surrounded by many cells and they themselves look like this. Okay, listen, suppose this is your what, zona glomerulosa layer. Suppose this is your glomerulosa layer. Okay, suppose this is your glomerulosa layer and this is your fasciculata layer. Fascicle means rope like, therefore I am drawing like this. Don't think that I am drawing the lipid bilayer. Okay, zona fasciculata, and then reticularis. Reticularis means network like. Okay, that means the cells are looking like this. In the section, cells are looking like this. Clear? So now at first we will be talking about the zona glomerulosa layer. Actually we will be drawing zona glomerulosa layer, then zona fasciculata, then zona reticularis. Okay. So how to draw is this zona glomerulosa layer. Then we will be drawing zona fasciculata layer. Okay. In fasciculata layer, the cells are forming a column-like structure. In the reticularis layer, you will find these arrangements of cells. They are actually defined. The cells are not present in a defined way, but rather present in a reticular formation. That is, they are forming a net-like structure. Okay, net of cells. Clear? They are forming a net of cell. Okay. So this is what? This is your zona glomerulosa. Clear? This is your zona fasciculata. This is your zona reticularis. You must understand this very basic thing. Okay. So this is these are the cells. Now listen with all your attention. You will actually when you will be studying all these things, you will remember this for your entire life, but right now you must understand, or you must at least know that these two layers perform similar actions, because these two layers, that is zona fasciculata and zona reticularis, these two layers have similar type of enzymes. They have similar type of enzymes, but the enzymatic difference lies in the zona glomerulosa layer. Therefore, you must understand that zona glomerulosa produces mineralocorticoid. Now, if you don't know what is mineralocorticoid, now listen. If I tell you you are a human, who are watching this video, you are a human. It's right? But you have a specific name, right? Like that. Mineralocorticoid is a big term. It's an umbrella term. And under the mineralocorticoid, there is a special mineralocorticoid which you all know, and you have already studied it. That is aldosterone. Aldosterone is actually your name, and mineralocorticoid is actually telling that you are a human. And a mineralocorticoid and aldosterone, they are actually the same, but they have a basic difference. All mineralocorticoids are not aldosterone. Clear? Now listen, this layer, that means these two layers combined, produces two hormones, two steroid hormones. Number one, glucocorticoid. Number two, sex corticoids. Number 1 mineralocorticoid, number 2 sex corticoids. Okay, clear? Now listen, you must understand again one thing. You know, there is something called adrenocorticotropic hormone, ACTH. There is something called ACTH or adrenocorticotropic hormone. Adrenocorticotropic hormone. Now listen, adrenocorticotropic hormones means what? Trophic means what? Health. Just understand this very basic thing. Trophic means what? Nutrition, right? That means this hormone, adrenocorticotropic hormone, this hormone actually maintains the health or nutrition or nutritional needs of adrenal cortex. Now, my question directly to you is, those who are watching the videos as well, my question directly to you is, adrenocorticotropic hormone, does it work in all these areas or all these zones? Or it works in special zones? Now listen with all your attention, many people don't know this. Adrenocorticotropic hormones have direct effect on these two areas. They control the secretion of steroid hormone from zona fasciculata and zona reticularis. They don't have any direct action or indirect action on zona glomerulosa. Now why I am saying this? Now listen with all your attention. There was a study where one of the patient had his hypothalamus removed, and therefore ACTH will not be secreted, because ACTH obviously is not secreted by hypothalamus, it is secreted pituitary, but it is controlled by corticotropic releasing hormone or CRH, and CRH actually activates the cells which secrete ACTH. Now when there is no CRH, there will be no ACTH, and therefore in these patients doctors saw that these two layers actually atrophies. These two layers, that is zona fasciculata and zona reticularis, only atrophies, but there is no change, histological change in the adrenal cortex zona glomerulosa layer. Clear? Clear everyone? Okay. Now we will be talking about the glucocorticoid chemistry. Okay. Now listen, before we start glucocorticoid chemistry, we want to tell you actually that what are the points upon which our discussion will be occurring. Now listen, we will be talking about, actually there will be 3 points. There will be 3 points. Actually it's a 3-point discussion. Okay, it's a 3-point discussion. What are those points? Number 1, steroid hormone synthesis. Steroid hormone synthesis and its basic biochemistry. Number 2, we will be talking about glucocorticoid signaling, and finally we will be talking about glucocorticoid function. Clear? So these are the 3 points upon which our whole lecture will be now occurring. Clear? If you really want to learn what is steroid hormone biochemistry, first understand or first try to learn the structure of cholesterol. Clear? Now listen, it's a very much basic for your understanding. Now, so if you want to really learn cholesterol structure, understand this very basic thing. Now, what is cholesterol? Now listen, cholesterol is one of the parent. Cholesterol is the parent steroid nucleus. Okay, what is the role of cholesterol? It's a parent steroid nucleus. It's a parent steroid nucleus. Okay, that means steroid hormones are actually nothing but the reorganized modeling of or model structure of cholesterol only. Clear? So you must understand what is a cholesterol and how it looks like. Now listen, first understand this very basic thing. If you are new to this biochemistry class, please don't panic. Now listen, first understand this very basic thing. Clear? Have you noticed one thing, that every steroid hormone ends in testosterone, progesterone, estrogen, right? Right? That means what? More often steroid hormones ends with ONE, right? Progesterone, estrogen, okay, or what, testosterone, that means what, they are actually ketones. They are actually ketones. Understand this very basic thing. They are biochemically, they are ketones. But if you try to study cholesterol, understand this very basic thing. At first you must understand cholesterol is an alcohol. It's an aromatic alcohol, not aromatic, but organic alcohol. Clear? It's an organic alcohol. That means how can you convert an alcohol into a ketone? By oxidation. That means cholesterol undergoes oxidation, no doubt about this. In majority of the cases, cholesterol undergoes oxidation. You will see that. Clear? The first thing you must be clear about. That means in your body, cholesterol undergoes oxidation. First thing. Let's talk about this now. How to draw a cholesterol? Okay. Now listen. So you want to learn how to draw a cholesterol. Now listen with all your attention. Draw with me. Okay. You must draw with me to understand. Okay. Now listen. There are 4 nucleus in a cholesterol. What are nucleus? Nucleuses are these things. Okay. So there are 4 nucleus. Now listen. So there are 4 nucleus, that is numbered A, B, C and D. Clear? There are 4 nucleus. Now listen. This is what? Hexagonal ring. This is hexagonal ring. This is hexagonal ring, and this is what? Pentagonal ring. That means cholesterol have 3 hexagonal and 1 pentagonal ring. Understand this first. Clear? Next. Okay. Now listen with all your attention. Listen. You are seeing these grooves, right? This is one of the groove, right? This is another groove. Okay. Clear? There are 3 grooves actually. Now listen. This is the first groove, this is the second groove, and this is the third groove, right? Now listen. In the first and the third groove, which are present on the upper side, there are actually 3 grooves in the lower side as well. But in the upper side grooves, in the first and the third groove, add a methyl group. Now you know why I have drawn like this. That means in the end there is methyl group. Clear? But no need to draw this. It will make things more complicated. Okay? Add methyl group. So in the first and third groove upside, add what? CH3 group. That's it. It's so simple. You have added it. Now listen with all your attention. Now add a double bond in this position of B, chain. Add double bond in B. Clear? B nucleus. Next. Add this chain to this portion. Okay. Now listen. Now we will be talking about so what is cholesterol? It's an alcohol, right? So here it is, the alcohol group. Clear? Here it is, the alcohol group. Now listen with all your attention. This is the basic structure. Okay, we have not completed it yet. Now listen. Let's start numbering. Okay, let's start numbering. Now listen. This is actually the structure of cholesterol but not glucocorticoid. Now listen, let's start numbering. Numbering starts from nucleus A, from this position. Okay, this is 1 2 3 4 5 6 7 8 9 10. Again, now listen, you will have to come like this. Okay, starting from here, this arrow will go like this, but after completing 10 you will move from here to here. That means start from here, then go to B, then up to 10. That means these two hexagonal rings have 10 carbons. Okay, then switch from here to here. Okay, from here to C. So this is 11 12 13 14, not here, this is 13, carbon number 13, 14 15 16 17. Okay, now listen, a very important point you need to understand. Now after you have completed numbering the nucleus, that is A, B, C and D, start numbering these branches, not this branch. This is 18, this is 19. Clear? Now listen, from 19 you will have to move to this junction, that is 20. Now listen, 21 lies here in the outside. 21, 22, 23, 24, 25, 26, 27. Clear? That means cholesterol have 27 carbon atoms. Have 27 carbon atoms. They have what? They have 27 carbon atoms. Clear? They have 27 carbon atoms numbered like this. Okay. Now, so this is the basic structure of a cholesterol molecule. If you really want to understand how this cholesterol is converted into a glucocorticoid or cortisol specifically, a very important point is this, that what I will be doing, I will be drawing this cholesterol structure here in a small manner so that I can remember the carbon numbers. Okay, you can also do this, and those of you who are watching the videos, follow the steps. What we are doing is we are actually making a short replica, small replica of this particular cholesterol structure here. Okay, so you take one copy and pen or whatever it is, whatever with you can be satisfied with, then you try to draw this whole biosynthesis of cortisol with me. Okay, try to solve this. So we all know that for cholesterol synthesis, for cortisol synthesis or glucocorticoid synthesis, we need what? We need what? We need cholesterol, right? We need cholesterol. Everybody knows that. But before you jump onto this conclusion that cholesterol can be converted into cortisol in an easy way, wait for it. There are multiple steps available. There are multiple steps available and they are not at all easy steps. Clear? But before we jump into the steps, you must understand this very carefully, that you will have to deal with cholesterol. That means either it is produced in your own cells. That means what? Your zona glomerulosa or zona fasciculata and zona reticularis, these cells are producing cholesterol, in situ cholesterol, and your body can actually utilize them. But listen with all your attention. These cells, zona glomerulosa even, glomerulosa, zona fasciculata and zona reticularis and all other organs in your body produces a very little amount of cholesterol. Therefore, those who are even taking atorvastatin, they also get sometimes heart attack, because they think that we are taking anti-cholesterol or anti-cholesterol agents or statins. That means we are safe. No, our body actually produces a very little amount of cholesterol. Most of our cholesterol comes from our diet. Remember this for your entire life. Most of our cholesterol comes from our diet. Our body produces, yes, they produces cholesterol, and it's a very lengthy process. There are 4 or 3 maybe Nobel Prizes associated with the discovery of cholesterol biosynthesis. We have talked about that. But remember this for your entire life, that our body actually produces a very minimal concentration of cholesterol itself. And most of our cholesterol, which is converted into either steroid hormone or vitamin D, they are actually, we are getting from Diet. That's the first thing you need to understand. That means, suppose if I want to convert a cholesterol into a cortisol, then the first thing you need to understand is this: there is a high possibility that this cholesterol molecule is actually, this whole cholesterol molecule is actually coming from our diet. Then the question arises is how this cholesterol molecule now enters zona reticularis or zona fasciculata cells, right? Now listen, it's a very, very long process, okay? We'll talk about that in short, but it's a very, very long process. Now listen, you know that if you don't know how LDL and HDL are produced, go back to the videos, you can understand then. Now listen, you know that one of the best carrier of cholesterol in our body, one of the best carrier of cholesterol is what? One of the best carrier of cholesterol is LDL or low density lipoprotein. In our body, one of the best carrier of cholesterol is low density lipoprotein. Clear? In our body, one of the best carrier of cholesterol is what? Low density lipoprotein. That means those cholesterols which are coming from our diet, they are, you know, packaged into LDL particles, and these LDL particles will now move throughout your bloodstream, and wherever these LDL receptors are expressed, they will get attached to these LDL receptors, and cholesterol will now enter the cells where LDL receptors are expressed, right? Now listen with all your attention. So one thing you can understand, that zona glomerulosa, zona fasciculata and zona reticularis, all these three layers have high level of, you know, LDL receptors, right? They have high level or high, you know, expression of LDL receptors, okay? Now listen with all your attention, it is very important for you to understand. So we will be talking about LDL, right? Now listen, LDL is one of the what? One of the best carrier of cholesterol. Clear? They are one of the best carrier of cholesterol. One of the best carrier of cholesterol is your LDL. Now a very important point here we need to understand is this, that suppose your LDL, suppose this is your LDL, clear? This is your covering of LDL molecule, clear? Now listen with all your attention possible. Now inside the, you know, LDL molecule, free cholesterols are present in a very much, you know, low concentration, but cholesterol in a different form is present in high concentration inside the LDL particle, okay? So this is the what? LDL particle, okay? So what is that form? This is cholesterol ester. So cholesterol esters are present in high concentration in the LDL particle, clear? Cholesterol esters are present in the high concentration in the LDL particle. Now listen, now we will have to decorate LDL particles. Now listen, you know that suppose this is your, you know, phospholipid, and this is what? Fatty acid chains. Now listen, this is polar head, right? This is polar, this is non-polar, right? This is non-polar. A very important point here we need to understand is this, that these non-polar chains are actually directed inwards, but these polar chains are exposed on both sides. Why? Because this is what? Extracellular fluid. This is what? Extracellular fluid. Now extracellular fluid has many polar molecules, and therefore polar will now interact with polar, okay? Now listen with all your attention, a very important point you need to understand here is this. So this is suppose your LDL molecule. Now listen, your LDL molecules have lipid bilayer outside. Your LDL molecules have lipid bilayers. Though they are, you know, phospholipid head, polar head are extended outwards, okay? Though their phospholipid heads are extended outwards, but still they are immiscible in plasma, because plasma contains a very much good amount of water. That means what? Why I am telling you these things? Now listen, that means to swim in that water of plasma, your LDL molecule needs some very much highly polar molecules. They are polar, but not to the extent that they are highly polar. They are not highly polar, they are polar, okay, moderately polar, but they need highly polar molecule to swim across the blood because blood contains a high concentration of water. That means there must be some proteins which are present outside. Now listen, suppose this is a boat and the protein is like what? Protein is like that engine. Clear? It's suppose it is a boat for the transfer of cholesterol, ester and tags. Okay, for the transfer of cholesterol, ester and tags, no doubt about this. Okay, but you need a boat, right? And the boat is LDL, but these boats should be supplied with what? Proper engines, right? Now these engines are actually, you know, these type of molecules. Clear? Now these engines have a very, very important impact on your, you know, health. Now listen, these engines, these protein engines which are polar, they are called apolipoprotein. They are called apolipoprotein. They are called apolipoprotein or simply apo. Clear? If you remember that in LDL, apolipoprotein B is present. It's a special type of apolipoprotein which is present only in LDL or low density lipoprotein. Clear? So this is what? This is ApoB. This is what? ApoB. This is what? ApoB. Now listen. So these are what? Apolipoproteins. Now a very important and interesting fact is this. If you have mutations in apolipoprotein, then your body will not be able to metabolize this LDL, and this LDL will be, you know, present in high concentration in your blood, and therefore you will suffer from a disease called hypercholesterolemia. Okay, and in hypercholesterolemia what happens is this, that people die of cardiac diseases. Clear? At an early age. Clear? Okay, now listen with all your attention why I was talking about these things. Okay, now this LDL and ApoB is actually the carrier of cholesterol. Okay, now listen with all your attention. Suppose this is your adrenal cortical cells. Suppose this is your adrenal cortical cell. Clear? That is zona fasciculata or zona reticularis specifically. So this is what? So this will be your phospholipid bilayer, right? This is your phospholipid bilayer. What we are actually trying to learn is this, that how these LDL receptors are, or how this LDL molecule are internalized by cells of zona glomerulosa, zona fasciculata. Clear? You know that these cells have high concentration of what? High concentration of LDL receptors. Now listen with all your attention. LDL receptors exactly look like this. Okay, LDL receptors looks like this. Now listen, a lipoprotein, an low density lipoprotein receptor or LDL receptor looks like this. Clear? Sorry, let me not point this part only. So this is what? This is your LDL receptor, right? This is your LDL receptor. Clear? Now a very important point here we need to understand is this. So this domain of LDL receptor, this portion is a very important domain. You know what is called? NPXY domain. Clear? It is NPXY domain, or it is also known by the name of sorting domain. Now why it is called sorting domain? Now listen, if you don't know these proteins are called adaptor protein 1 or AP1. Okay, these proteins are called adaptor protein 1 or AP1. Now you know what is the importance of this AP1? Do you know what is the importance of this AP1? Now listen, this AP1 is very important because this AP1 actually is associated with another protein. Listen, this AP1 is associated with another protein like this, okay, and this protein, this one is called clathrin. Now you know what I am drawing, that is, so this is what? This is clathrin. Now listen with all your attention. Every AP1 have connections with clathrin. Clear? Every AP1 has connections with clathrin. Now what happens is this, that when LDL binds with, you know, LDL receptor, then clathrin mediated endocytosis takes place. Clathrin mediated endocytosis takes place. We will talk about this in details when we will be talking about molecular biology of the cell. Clear? Now listen, clathrin mediated endocytosis takes place. Now it is very important for you to understand is this, that if this domain, this NPXY domain is disturbed or is mutated, then this particular domain never binds with this AP1 or adaptor protein 1, and therefore also you may have hypercholesterolemia due to high concentration of LDL in your blood. Now listen, why this domain is called NPXY domain, okay? Why this domain is called NPXY domain? Now N stands for what? You know, asparagine. These are nothing but what? Shorthands of your one letter code for your amino acid. P for proline. X for, you know, what do you mean by X? X for any amino acid other than this, or may include this, any amino acid, and Y means, you already know, tyrosine. So that means whenever you are talking about this NPXY domain, it directly indicates that this NPXY domain should have these 4 amino acids, okay, or to be very specific, asparagine, proline, any amino acid, or tyrosine. This sequence, NPXY sequence, should be present in this protein up to this level, okay, up to this level. And if there is a mutation which results in disturbance of this, you know, particular sequence, then this particular domain cannot bind with AP1 or adaptor protein 1, and therefore hypercholesterolemia occurs. Clear? Clear? Now listen, it is very important for you to understand. Now if this domain, suppose this is a protein, this whole, you know, this whole thing is a protein, okay. Now let me tell you about this thing first, okay. Now this is an NPXY domain of LDL receptor, and this is what? This is your beta propeller domain. This is called beta propeller domain. This is called beta propeller domain. Clear? And this is the sequence. Now listen, count this. Count each, red balls. Why are you laughing hearing the term ball? You are interested in balls? Now listen. How many are there? There are 1, 2, 3, 4, 5, 6, 7. Now listen. These 7, now listen with all your attention. These 7 amino acids, these are amino acids, okay, are all cysteine. They are cysteine rich residues, okay. These are what? All are this is what? This is what? These are cysteine rich domains. Okay, now listen with all your attention. These cysteine rich domains actually interact or bind with apolipoprotein B. Okay, this cysteine rich domain actually binds with apo B. Clear? Cysteine rich domain binds with apo B. Clear? Okay. So this is the overall structure and detailing of LDL receptors. Okay. Now we will be talking about how this receptor interacts with LDL and what happens to this LDL and how cholesterol are formed from cholesterol esters which are present inside this particular what? LDL. Clear? Let's start. Okay. So this is your phospholipid bilayer. Now listen with all your attention possible. Now tell me what is this? This domain of LDL receptor is called NPXY domain, right? Now to this what is attached? To this what is attached? What are these? This is AP1, right? This is AP1. This is AP1. Okay. Now this is what? This is your beta propeller domain, right? And to this 7 cysteine residues are present which will interact with 1, 2, 3, 4, 5, 6, 7. So there are 7 residues which will interact with what? Apolipoprotein B, which is present on the surface of LDL or low density lipoprotein. Okay. So this is suppose your LDL molecule. Now let me draw like this. Suppose this is your LDL molecule. So this is your LDL molecule. We need a bigger board, no doubt about this. Suppose this is your LDL molecule, and suppose this is your, this yellow one is your what? Apolipoprotein B, right? This is apo B, okay, and this is what? LDL. Clear? Now what are these? What are these? These are clathrin, right? Now listen, when this cysteine molecule, the cysteine residues binds with apolipoprotein B or ApoB, then internalization of this whole thing takes place. Internalization of this whole thing takes place. That means, you must understand this, that means internalization means this, the second diagram. If you want to really learn these things, draw with me. So internalization has actually started, and clathrin mediated endocytosis is taking place. This is what? AP1, right? This is AP1, and these AP1s are connected to LDL receptors 1, 2, 3, 4, 5, 6, 7, right? And here it is. Here, so this is apolipoprotein B, ApoB, right? This is ApoB. So now listen. So you know what is early endosome? Now listen, this is an endosome, but this is not a proper endosome. This is not a proper endosome. Why? Now listen, if you want to really learn what is endosome, actually endosome, this term is applied when this clathrin is absent. Now listen, when this clathrin is absent and AP1 is also absent, when this clathrin and AP1 is absent, this is called early endosome. This is what an endosome looks like. Now you are confused. Why I am telling? Because you are thinking that what is this? If you don't have any AP1 protein, then how this particular thing will be in place, right? Now listen, AP1 and clathrin is especially important for endocytosis, not for placing these things, not for placing this, what? LDL receptors, okay? So you must understand, so this is what? This step 1 is actually endocytosis, right? So you must understand that this is all what? In the zona glomerulosa, zona fasciculata, zona reticularis. This whole thing is occurring in these layers, okay? So what is this? This is early endosome. Why it is early endosome? Because there is no what? AP1 and clathrin, okay? When this early endosome is attacked by, a lysosome, now what happens is this, lysosome have in them, some acidic pH, lysosomes have actually enzymes which act in acidic pH. Therefore, the internal, property of lysosome is they actually maintain a low pH, okay? They have what? A low pH, right? Now a very important point, when this lysosome attacks this early endosome, then this whole thing, when this thing attacks, that means suppose this is what? Suppose this is your what? This is what? Lysosome plus endosome, right? Now when this scenario appears, this lysosome plus endosome is actually called late endosome, is actually called late endosome, is actually called late endosome. Clear? This lysosome plus endosome is called late endosome. Now listen with all your attention. When this lysosome interacts with or fuse with endosomes or early endosomes, then they will release what? Acidic enzymes, right? Hydrolysis. Now listen with all your attention. When these acidic enzymes are released, then this whole thing becomes acidic, and due to this decrease in pH, there will be change in this particular LDL receptor, and therefore what will happen is, in this case, suppose this is late endosome, that means when these things have integrated themselves. Okay, now this is what? Late endosome. This is what? Late endosome. Now listen with all your attention. When acidic enzymes interact with this particular engagement of low density lipoprotein and low density lipoprotein receptors, then this particular attachment is disrupted, and low density lipoprotein receptors actually release the low density lipoprotein particles. And what happens is, this is very important, and what happens is, these low density lipoproteins are actually now free from, so this is what? Apolipoprotein B or ApoB. Low density lipoprotein receptors are actually now free. Clear? They are now free to interact with other apolipoprotein, which are need 1 2 3 4 5 6 7. Okay. Now this thing will now go back to its original place. That means they will again place themselves in the cell surface membrane, and this thing will now be destroyed by enzymes, by the enzymes which are secreted here, and these enzymes will now release, cholesterol ester, a small quantity of cholesterol plus tags into this compartment. Okay, now listen. These cholesterol esters are actually the main sources of cholesterol in the formation of glucocorticoid, okay, or cortisol. So these cholesterol esters should be converted into cholesterol. Clear? That means what we will have to do? These cholesterol esters, to convert these cholesterol esters into cholesterol, what we will have to do? We need cholesterol esterase, right? Cholesterol esterases. That means what? They will actually release this fatty acid groups from cholesterol, and we will be getting cholesterol out of this cholesterol ester, right? So now by this time we know that how actually cholesterol molecule enters zona fasciculata and zona reticularis cells. Okay, now we will be talking about what is the fate of this cholesterol and how this cholesterol actually is converted into a glucocorticoid at a cortisol. Okay, now listen. So suppose this is a cell. Clear? Now this is your cholesterol ester, right? So this is your cholesterol ester, right? Clear? Now how a cholesterol ester looks like, now listen. So suppose this is your So suppose this is your cholesterol and suppose this is your suppose this is your cholesterol ester. Okay, now what we will have to do is we will have to actually cut down this portion and we will have to introduce an H here and an OH here. That's it. That means we will have to introduce H2O molecule to this particular bond. Okay, that's it. So this is done by cholesterol ester hydrolase. This is done by a enzyme called cholesterol ester hydrolase. Okay, cholesterol ester hydrolase. Okay, now this cholesterol ester hydrolase acts on cholesterol ester and this cholesterol ester then is converted into fatty acid and cholesterol. Okay, is converted into fatty acid and cholesterol. Now what will happen to this cholesterol? So this is what zona fasciculata or zona reticularis layer or cell, sorry, reticularis cell. Okay, so what will happen to this cholesterol right now? This is our important question. Okay, now this cholesterol through multiple pathways which is occurring in multiple organelles, through multiple pathways will be converted into cortisol. We'll only talk about this conversion of this cholesterol into cortisol. That's it. Clear? Now let's talk about this steroidogenic pathway. Okay.